In laboratories around the world, a scientific revolution is quietly taking place. Researchers are no longer just looking at cells under microscopes, they're listening to them. Welcome to the world of biosonification, where complex biological data is transformed into sound, revealing patterns and insights that visual analysis might miss. Today, we'll explore how this innovative approach is changing our understanding of biology, enhancing medical treatments, and even connecting modern science with ancient wisdom traditions. In a laboratory at the University of Padua, Italy, Dr. Alexandra Suvorov sits with her eyes closed, listening intently to what sounds like an avant-garde musical composition. But this isn't the work of a human composer. The haunting melody is actually the sound of a pancreatic cancer cell undergoing apoptosis, programmed cell death. As the rhythmic patterns suddenly shift, they reveal a cellular process that might have gone completely unnoticed through visual analysis alone. The human ear can detect patterns the eye might miss, explained Suvorov. When we translate biological data into sound, we leverage an entirely different cognitive pathway for scientific discovery. This emerging field represents a radical departure from traditional data visualization techniques. While converting data to sound isn't new, the sophisticated application to cellular biology has only gained serious traction since 2019, with researchers discovering that our auditory systems can identify subtle patterns in complex biological processes that visual representations often obscure. The technique has evolved significantly from earlier sonification efforts. Modern biosonification maps multiple cellular parameters simultaneously, from membrane potential fluctuations to protein synthesis rates, creating rich, multidimensional soundscapes. These sophisticated audio translations reveal the dynamic interplay of cellular processes in ways traditional visualization cannot match. At its core, biosonification acknowledges a fundamental truth. Cells are not static entities but dynamic systems in constant flux. The rhythmic nature of cellular processes, from calcium oscillations to circadian gene expression patterns, lends itself naturally to musical interpretation. One of the most powerful aspects of modern biosonification is its ability to compress timescales, allowing researchers to perceive patterns that might unfold over hours or days in just minutes of listening. This temporal compression gives scientists an unprecedented ability to detect subtle changes in relationships within cellular behavior that might otherwise require weeks of analysis. Traditionally, scientists have relied heavily on visual representations, graphs, models, and microscopy images to understand biological data. However, the human visual system has significant limitations when processing multidimensional datasets. Dr. Mark Ballora, a pioneer in sonification at Penn State University who passed away in 2019, argued that our culture is visually dominant but orally deficient. His groundbreaking work demonstrated that sound could represent up to 20 variables simultaneously, while visual graphs become unintelligible beyond 3 to 4 variables. This fundamental insight has transformed how researchers approach complex biological data. Building on Ballora's foundation, the Biorhythm Consortium, a collaboration between biologists, musicians, and data scientists across 9 countries, has developed standardized protocols for converting various biological processes into sound. These protocols ensure consistency and reproducibility in biosonification research, allowing scientists worldwide to speak the same audio language when discussing cellular behavior. The advantages of auditory data perception extend far beyond multidimensionality. Our auditory system excels at detecting temporal patterns and subtle variations in rhythm, making it ideally suited for analyzing the dynamic, time-dependent nature of cellular processes. Additionally, we can listen while performing other tasks, allowing for background monitoring of experimental data in ways that visual observation cannot accommodate. This multitasking capability significantly increases laboratory efficiency. Neuroscience research strongly supports this approach. Studies by Dr. Nina Kraus at Northwestern University have shown that the auditory cortex processes information in fundamentally different ways from the visual cortex, with particular strengths in detecting temporal patterns and rhythmic inconsistencies. By engaging these complementary neural pathways, biosonification effectively doubles the cognitive resources available for data analysis, creating new possibilities for scientific discovery. The process of biosonification involves several sophisticated steps that transform biological data into meaningful sound. It begins with gathering high-resolution temporal data from cellular processes using techniques like single-cell RNA sequencing, calcium imaging, or real-time metabolic monitoring. These datasets often include thousands of variables tracked over time, creating incredibly rich material for sonification. Parameter mapping forms the critical next step, where biological variables are assigned to specific sonic elements. For example, gene expression levels might control pitch, protein interaction rates determine tempo, membrane potential influences timbre, metabolic activity governs volume, and spatial movement affects stereo positioning. These mappings aren't arbitrary but designed to create intuitive relationships between biological significance and perceptual prominence. Sonification algorithms have evolved dramatically in recent years. Early approaches used simple direct parameter mapping, but modern systems employ machine learning to identify the most perceptually significant patterns. The SoniCell platform, developed at the Max Planck Institute for Biophysical Chemistry, uses neural networks trained on both biological datasets and music perception research to generate sonifications that highlight biologically meaningful patterns while remaining aesthetically coherent. Dr. Hiroshi Nakagawa's open-source CellTone platform represents another significant advance in the field, incorporating psychoacoustic principles to optimize the perceptual clarity of the resulting sounds. What makes our approach unique is that we're not just creating arbitrary sounds from data, Nakagawa explains. We're designing sonifications that respect both biological relevance and musical coherence. This balance between scientific accuracy and auditory clarity is essential for effective biosonification. The technical infrastructure supporting biosonification has also matured significantly. Specialized audio hardware with 128-channel capability now allows for the spatial representation of subcellular components, creating immersive 3D soundscapes that correspond to cellular architecture. Meanwhile, real-time processing systems can now sonify data streams with less than 10 milliseconds of latency, enabling truly interactive exploration of biological datasets as they're being generated. Beyond its value as a research tool, biosonification is showing remarkable promise in clinical settings. At Memorial Sloan Kettering Cancer Center, oncologists are now using sonification to monitor real-time treatment responses in cancer patients. Dr. Leila Perhaji, who leads the Clinical Biosonification Initiative, describes a recent breakthrough. We sonified the metabolic profiles of glioblastoma cells during treatment with an experimental drug. The oncologist could literally hear when the cells developed resistance. It sounded like a key change in music, 3 hours before conventional biomarkers showed any change. This early detection allowed for rapid adjustment of treatment protocols, potentially adding months to patient survival times. The immediate auditory feedback provides clinicians with crucial information at speeds that traditional monitoring methods simply cannot match, potentially transforming how we approach cancer treatment monitoring. The therapeutic applications extend far beyond monitoring. At the University of California, San Francisco, researchers in the Cellular Acoustics Laboratory are exploring how specific sound frequencies might directly influence cellular behavior. Their preliminary work suggests that playing specific sonification patterns back to cancer cells can actually slow their proliferation rate, a phenomenon they've termed acoustic entrainment. This builds on earlier work in mechanobiology showing that physical vibrations can affect cellular signaling pathways. Dr. James Collins at MIT has demonstrated that mechanical stimulation at specific frequencies can alter gene expression patterns in mammalian cells. Biosonification provides a sophisticated means to translate these insights into precisely targeted acoustic therapies. The implications are profound. Sound patterns derived from cellular processes might eventually be used to modify those very processes, creating a feedback loop with therapeutic potential. In surgical settings, biosonification is finding applications in real-time tissue assessment. Surgeons at the Cleveland Clinic now use a handheld probe that sonifies the metabolic activity of tissues during tumor resection procedures, helping to distinguish between healthy and cancerous tissue with greater precision than visual inspection alone. In an unexpected cross-disciplinary development, ethnomusicologists have noted striking similarities between certain cellular sonifications and indigenous musical traditions. Dr. Eduardo Montero, a molecular biologist and ethnomusicologist at the Federal University of Minas Gerais in Brazil, has documented parallels between mitochondrial energy production cycles when sonified and traditional Bororo rhythmic patterns from central Brazil. The Bororo have songs they describe as the sound of life continuing, Montero notes. When we sonified the electron transport chain, the process that generates cellular energy, the rhythmic structure was remarkably similar to these traditional songs. It raises fascinating questions about how ancient cultures might have intuited biological patterns. This connection extends beyond mere coincidence. Several indigenous healing traditions incorporate rhythmic sound as a therapeutic element. The Shipibo-Conibo people of the Peruvian Amazon use icaros, healing songs that are said to represent the energetic patterns of medicinal plants. When researchers sonified the molecular vibration patterns of compounds from these plants, they found structural similarities to the traditional melodies. These findings suggest a profound possibility that traditional knowledge systems may have developed ways to perceive and work with biological rhythms that modern science is only now rediscovering through technology. Perhaps the most revolutionary aspect of biosonification is its potential for democratizing complex biological data. The SonoCell project, launched in 2021, allows anyone with a smartphone to listen to sonified cellular processes through an augmented reality app. When pointed at printed cellular diagrams in textbooks or scientific papers, the app generates real-time sonifications based on the latest research data. More than 30,000 high school and undergraduate students now use the platform. Students who are blind or visually impaired can now hear cell biology in unprecedented detail, says Dr. Vera Koklova, SonoCell's lead developer. But we found that all students benefit. Sound creates an emotional connection to abstract concepts that visual representations often fail to establish. This accessibility extends to citizen science initiatives as well. The Cellular Symphony project invites participants to contribute recordings of their sonified biological data, creating a growing library of cellular sounds. Machine learning algorithms then analyze this collective database to identify patterns across cell types and conditions. This innovative approach has already identified previously unknown rhythmic signatures associated with early-stage neurodegenerative processes. Educational applications continue to expand rapidly. The Bioacoustic Lab program, now implemented in over 200 high schools globally, provides students with simplified sonification tools to explore biological datasets. This hands-on approach has shown remarkable results in engaging students typically underrepresented in STEM fields, with participation rates among female and minority students significantly higher than in traditional biology laboratory courses. The multisensory approach seems to break down barriers to scientific engagement that have persisted for generations. Students report that hearing cellular processes makes biology feel more relevant and accessible than traditional textbook approaches. The emotional engagement with sound creates memorable learning experiences that persist long after the class ends. This educational revolution extends beyond K-12 education into medical training as well, where sonification is helping medical students develop more intuitive understandings of physiological processes. Beyond its practical applications, biosonification raises profound philosophical questions about our relationship with the microscopic world and the nature of life itself. When you listen to a cell division sonification for the first time, something changes in your perception, says Dr. Nakagawa. These processes inside us are no longer abstract concepts. They become experiential. You realize you're made of trillions of tiny musicians playing an incredibly complex symphony. This shift in perspective challenges the mechanistic view of cellular biology that has dominated scientific thought since the Industrial Revolution. Instead of cells as tiny machines, biosonification suggests a more dynamic, rhythmic understanding of life, one that resonates with both ancient wisdom traditions and contemporary complex systems theory. This philosophical reframing has implications beyond science, potentially influencing how we conceptualize our relationship with our bodies and the natural world. Looking forward, several emerging directions promise to expand the field even further. Multicellular orchestration, sonifying interactions between different cell types simultaneously, reveals emergent properties in tissue systems that weren't apparent through other analytical methods. Researchers at Zurich have created tissue symphonies, demonstrating how cellular communication creates higher-order patterns across biological scales. Diagnostic applications continue to advance rapidly, with machine learning systems now able to identify disease-specific sound signatures in sonified patient samples with accuracy rivaling traditional laboratory tests. The startup Audible Health is developing a platform that can screen for multiple conditions simultaneously by analyzing the acoustic patterns in sonified blood samples. This could revolutionize early disease detection, particularly in resource-limited settings. Perhaps most intriguing is the potential for biosonification to bridge the gap between scientific understanding and public engagement with biology. As Dr. Suvorov reflects, we've spent centuries trying to understand life by looking at it. Perhaps it's time we also listened. The emergence of biosonification represents more than just a new scientific technique. It embodies a fundamental shift in how we perceive and interact with the living world. By translating the complex dynamics of cellular processes into sound, researchers are not only discovering new patterns and relationships invisible to the eye but also reconnecting with ancient understandings of life's inherently rhythmic nature. From cancer treatment rooms to high school classrooms, from surgical theaters to indigenous healing ceremonies, the ability to hear biology is creating bridges between disciplines, cultures, and ways of knowing that have long been separated. As we continue to develop this multisensory approach to science, we may find that the symphony of cells playing within and around us holds answers to questions we're only beginning to ask. In this listening, we discover not just new scientific insights, but a renewed sense of wonder at the musical nature of life itself, a symphony playing at scales both infinitesimal and vast, connecting us to the rhythmic processes that have sustained life for billions of years. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.